Okay, so welcome to the afternoon session, or the last session of the day. So our next speaker is Enzo Marsot, and he's going to talk about neural networks and how they can predict ghost species diversity from gene transfers.
I'm just waiting for people to. Okay. So in this talk, I'm going to talk about ghost species and how we can obtain information about them. So we will use a neural network approach and leverage horizontal gene transfers to obtain information about ghost species. So first, I'll introduce what ghost species are, then introduce rapidly horizontal gene transfers, explain how we can use the signal from horizontal gene transfers to estimate the amount of ghost species. And finally, I'll present a proof of concept on simulated data of the possibility to recover ghost diversity from horizontal gene transfers. So, as a motivation, most of the biodiversity, actually the overwhelming proportion of biodiversity is now believed to be extinct, and most species of many groups are unknown today, even though they are still living. And many of these species, when they go extinct, they do not leave fossils or genetic information for us to know they existed or for us to characterize them. But they may have transferred some of their genetic material to species whose genome we know today. So yeah, we could use this information to obtain their diversity, ghost biodiversity. So this is the problem setting. Let's suppose you have a complete tree. So this represents the set of all species we consider. There are extinct species like G and F. This is a current time, the dotted line. The extant tree is the species that are still alive today, and the sample tree is the subset of species that are alive today, so we can sample their genome, and that we know. So our goal is to obtain information about the whole tree from just this, which doesn't seem obvious, but I'll explain how we can do it. So horizontal gene transfer is the movement of genetic material between organisms in a manner other than traditional reproduction. So you can imagine like two bacteria, one of which just sends one of its gene in some box or something to another bacteria. And it's actually very important for bacteria because it is a factor of resistance to antibiotics. So in this talk, we will talk about horizontal gene transfers with replacement. So if you have two species with one version of the same gene for each, the transfer of species A to species B will replace B's version of the gene. So let's see how we can see that a horizontal gene transfer occurred. So in this example, we suppose we have this species and a transfer occurs here. Now, it means that when we get the genome of A and B, for the gene G, they will look way closer than they actually are as species. So we obtain this discrepancy between the species tree and the gene tree. So we actually have algorithms which allow us to go from this discrepancy from G and the species tree to a transfer explaining the discrepancy. So in this case, that's okay. We suppose there are no ghost species and everything functions fine. Now let's suppose we introduce a ghost species G. So G can also send its version of a gene to the species B, which we know. So this will be the tree we know. And after the transfer from G to B, B will look close to this species, so we will obtain this gene tree for the species A, B, C, D we know. So there are two remarks we can make. First, there is a discrepancy, as in the previous example, between the species tree and the gene tree. But this time, if we reconcile the topologies of the two trees, we will actually infer a transfer between this species, so this one, and B. But this is impossible because they have never lived together. So the fact that a species sends a gene to a non-contemporaneous species may indicate that there are ghosts. Yeah, another factor is the length of the branches of the gene tree. So in this case, they become shorter than they would have been. The gene tree would have this shape if there were no transfer, but now the branches are shorter. If it's a transfer from a ghost species, the branches become longer. So I make these observations to show that there are actually signs that a transfer comes from a ghost species and not just from a species that is actually within the tree. Another observation which was made by Theo, who helped me on this project, is that if we suppose there is a bunch of species here that just went extinct, and they send transfers to this tree which we know today, the transfers coming from this part of the tree will actually be attributed to this one. So we will attribute an excess of transfer from this tree, whereas given its length, we would not expect it to send so many transfers to the rest of the tree. So to try to approach this problem, we simulate species tree, horizontal gene transfers, and sample leaves. So this creates ghosts. This example, our sample tree will be this, and the ghosts will be basically G, F, and E. And we train a neural network to predict ghost length. So what we call ghost length is like A here still exists here, but E doesn't exist, F doesn't exist, and G doesn't exist. But you attribute the sum of these lengths, this one, this one, this one, and this one, to A. So that's what you're trying to predict for A. We then complexify simulations varying various parameters and the network architecture simultaneously. So what we need to do is take into account the inductive biases of our problem, the properties of our problem. So we are working on graphs, more specifically trees. So we need our model to be equivariant with respect to permutation of the nodes. If we just change the representation of the tree, we should not obtain a different result. That's why we use the graph neural networks. And there are several approaches for us to make the problem equivariant. the simplest is to treat it node by node. So you just take a node of the species tree, try to predict the ghost length without looking at the rest of the tree. You use just summary statistics like depth, the number of transfers received and donated, transfers to the future or descendant, which we said were important since transfers to the future are actually impossible normally, but they can be inferred anyway if there are ghosts. The second approach is to use a graph neural networks along the horizontal gene transfers. So horizontal gene transfers are like edges of some graph. So you can do message passing along them. The last thing you can do is take into account like the phylogenetic tree graph structure. And to obtain an equivariant architecture, we went with transformers because they have had a lot of success first with natural language processing, with vision, and now with graphs. It's still state of the art in many cases. And we used the graph GPS, which was a state of the art at the time. This is the architecture. So we try to use a graph transformer architecture with a self attention, a message passing convolution along the graph of transfers and along the parenthood graph, and then we predict ghost length for each of the nodes. So our results showed that the more complicated the simulations, the more complicated the architecture needs to be to take this into account. So in this case, we took simulation parameters which were constant throughout the dataset. So there's always the same number of species. The number of species sampled is the same. The transfer rate for horizontal gene transfers is the same. So in this case, you can basically learn your data with like almost a linear regression. So sorry, it's in French. So this is mean absolute error on the prediction of ghost length. And this is the target ghost length for one branch. So let's take like this one. On average, it's like 20% error maximum of prediction, which is pretty good. In this case, however, we varied the parameters in the dataset. So the number of species can change between species tree. We sample different numbers of species. We change the transfer rates. And this shows something which is very interesting. Like take the linear and MLP architectures, which only work on a node one by one. It doesn't take into account the whole structure of the tree. So these architectures don't work well because if you only take into account the nodes one by one, you cannot infer parameters such as transfer rates or the number of species in the tree. So you cannot like correct your prediction with global parameters of the tree, which would be useful. So we have open questions after this work. There's lots of work we could still do to see what the limits of our approach are. So, for example, we could create ghosts by adding extinct species. In our case, we have only created ghosts by generating a species tree with a pure birth model, then sampling species from this tree. So there was no extinction. So we wonder if our inference still works when there's extinction. And one question is, like, we infer information about ghosts in the complete tree, but we only have sampled a proportion of the complete tree. So how small can this proportion be for us to still recover like information about the whole tree? Like if you have 6,000 species in total, but you only sample 6, it's obvious that your prediction of ghost length will not be very good because you don't have very much information. There's also room for further exploration, like modifying our pipeline, especially. The reconciliations we make on big trees to obtain the transfers, they take a lot of time. It really limits us. We have opportunities for experimental validation. So, for example, we could test our model on some tree of bacteria, just remove a group of species from this tree. So they will basically become ghost. It's like they have become ghost species. And if the prediction of our model increases the ghost length by the appropriate amount, then we are confident in its predictions. And we could try to find a trace of historical events such as mass extinctions on experimental data.